Thanks so much, Chris. So yeah, it's a great pleasure to present to you all today about the work that we've done on pushing the limits of gradient technology. And so I'll be showing some results highlighting extreme neuroimaging with the various Connectome scanners we've worked on, and then also a preview of what's to come. So as Camille pointed out, the fundamentals of MRI physics, I think, really tell us that there's more signal to be seen as the magnetic field strengths increase. And what I'd like to shine a light on today is how pushing the limits of our other magnetic field, our gradient fields, will allow us to encode spatial properties at all scales, both at the macroscopic scale for imaging and also at the microscopic scale on the level of cells and tissues by increasing the power and speed of those gradients. So this work that I'm presenting today is really built upon more than a decade of collaborations and collective efforts from the incredible scientists and engineers that I've had the privilege of working with at the Martino Center at Mass General in partnership with Siemens. And this was really first kind of sponsored and funded through the NIH, through the Human Connectome project, and then more recently through the Brain Initiative to really push the limits of MRI hardware and acquisition technology, and in particular for diffusion MRI. So why diffusion? Well, I would argue that diffusion amongst all the different MRI contrasts is unique in that it provides us with an indirect probe of structures at the micron level in tissue. So really getting to the scale of individual cells and axons by using the Brownian motion of water to help us infer their characteristics. And I'll point out that we're accessing information, kind of drilling into the voxel, so at that microscopic level, beyond the nominal voxel resolution of what MRI is typically able to achieve, which for clinical MRI is usually on the order of millimeters. So I would argue that diffusion MRI, amongst all the contrasts, holds the most promise for probing cellular structures of any depth and location in the living human brain. And this has led to a central vision that we have in microstructural imaging using diffusion of offering in vivo histology, allowing us to estimate and map the histological features of tissues in the living human brain using diffusion. So why do we need strong gradients for diffusion MRI? Well, in a conventional MRI scanner, typically with gradient strengths between 40 to 80 millitesla per meter, in order to achieve a strong diffusion encoding, diffusion weighting, we have to play out these diffusion encoding gradients for a long time. And that really pushes out the overall signal acquisition to longer times, incurring signal loss from T2 relaxation and overall diminishing the signal to noise ratio. So on the Connectome systems, these are systems that are able to achieve close to an order of magnitude higher in maximum gradient strength. The stronger gradient amplitudes really allow us to increase the efficiency of that diffusion encoding. And so essentially, we can reduce the amount of time needed to do the diffusion encoding. And this leads to a very straightforward benefit in terms of shortening the echo time and boosting the signal to noise ratio. What's perhaps less kind of well appreciated, but actually definitely a boon for diffusion imaging, is that using stronger gradient strengths sensitizes the measurement to smaller displacements based on diffusion, and that will really increase the resolution of structures at that microscopic scale. So it essentially improves the diffusion resolution. And then finally, the shorter diffusion times that we can access using these stronger gradients sharpens up the diffusion profile and allows us to, say, track between fibers, especially those with low crossing angles or at complex fiber configurations. So the original Connectome scanner, which was installed at the MGH in 2011, was really considered the ultimate diffusion machine when it was installed. It featured 7 times the clinical gradient strength with a maximum gradient strength of 300 millitesla per meter and a slew rate of 200 Tesla per meter per standard second, which is pretty standard. But alongside this sort of centerpiece gradient technology, this was really a massive gradient. It was a whole body gradient that was pieced together in 4 segments, each of which was powered by a gradient amplifier along 3 axes. in addition to having this very powerful gradient, which I think Larry Wald liked to make the analogy that it took the amount of power to power, say, a Los Angeles class nuclear submarine. I will say, based on anecdotal experience, it took about 10% of the power from the city of Charlestown when it was operating at maximum capacity. So really making full use of this massive gradient also kind of stimulated the innovation of our MR physics group to allow these measurements to be done in a feasible way to map structural connectivity within the whole brain. And so we developed acquisition advances through simultaneous multi-slice imaging led by Cowan Setzenpop and others that really allowed us to do full brain encoding, exciting multiple slices at a time within a reasonable scan time, allowing us to do whole brain measurements for diffusion. And that, of course, was coupled with high sensitivity multi-coil receive arrays led by Boris Kyle in our lab at the time. And so in aggregate, all of these advances enabled us to achieve nearly 10 times increase in sensitivity for high B-value diffusion. So what did we use those strong gradients for? Well, I think the goal of the original human connectome project was to map all the major structural connections in the human brain in sort of adult populations, and then went on to look at disease populations. And so what we used these gradients for was to improve the sensitivity of the diffusion encoded subvolumes that would then allow us to track through, basically get sharper definition of these water diffusion probability distribution functions, and then allow us to capture more of the fibers throughout the whole brain. This was demonstrated in an ex vivo macaque with diffusion spectrum imaging by Van Wedeen. We've gone on to do that through multiple healthy subjects and healthy cohorts, culminating in the MGH USC Human Connectome Project diffusion data set. What was less appreciated at the time when the scanner was installed is that these strong gradients could also be used for capturing the microscopic structure of tissues. And so what we've done over the last decade or so is to show systematically that you can map quantities like axonal diameter, essentially using these very strong gradients to access diffusion properties within very restrictive spaces, including within axons. And we and others, including here at NYU, have shown that these types of quantities are not really attainable with your conventional clinical gradient strengths up to 80 millitesla per meter. You're just not sensitive to that information. So here I'm showing the ability to map axonal diameter in the whole brain. We averaged this across 20 healthy subjects using standard sort of 2 millimeter voxel size. And what we were able to appreciate, especially as you average over subjects, is that there are some really interesting trends in axonal diameter that are captured in these maps that replicate what people have seen on neuroanatomical studies for decades, both in non-human primates and also in humans on autopsies. So for one, the motor tracts, the corticospinal tracts, seem to be much larger than the surrounding white matter. As we average over subjects, we can also see that there's this interesting anterior to posterior gradient of axonal diameters, where you see smaller diameter fibers in the frontal lobes, which are thought to be subserving the finer processing functions of the frontal lobe, and then larger diameter ramifications going to the parietal and occipital lobes, really serving the huge amount of information that is processed for the visual system. And so these results were in agreement with what others have shown from these complementary studies in non-human primates and also in humans. And so we were really encouraged by these results, but we also realized that there were some limitations to our studies. And that's really kind of spurred on our efforts over the last 5 years to push the envelope further on gradient technology for diffusion MRI in particular, and looking specifically at microstructure. So why do we need a next generation connectome, or connectome 2 as we've been calling it? Well, what we realized through systematic and careful theoretical studies as well as simulations is that higher gradient strengths and slew rates are needed to achieve even shorter effective diffusion times, and also to probe micron-sized structures at a smaller length scale than what we're currently able to access. As just one example, as our colleagues here at NYU know very well, axonal diameters are typically on the order of about a micron in size, yet the types of estimates that we were obtaining from our studies originally were actually on the order of 5 to 6 microns. And so there's clearly a bias that's present in these measurements, and we can think of these more as axonal diameter weighted maps as opposed to true axonal diameter. Another reason for and kind of a very important parallel effort that's been ongoing through the development of this technology is that we realize we need systematic multi-scale validation studies to inform better biophysical models of brain tissue microstructure. So keep in mind that these estimates of axonal diameter, cell size, cell shape, these are all inferred quantities. We're not imaging them directly with the MRI scanner. And so there needs to be fundamentally kind of a good understanding of how to bridge the gap between what we're seeing on MRI and what we see under the microscope. And then finally, if we're successful in achieving these estimates and kind of pushing the technology to where it can go, we believe that pushing the limits of in vivo diffusion MRI will enable us to see how the brain is changing in living humans as they experience different life events. So as they learn new skills, new tasks, as they capture the changes potentially throughout the pathology spectrum. So can we detect changes at that microscopic level that would manifest even before you see actual structural changes in the brain, say through cortical volume, cortical thickness changes, or white matter lesions? And so I'm really pleased to sort of share the latest results that we've gotten from the Connectome 2 scanner. The scanner was installed in July at the MGH. It achieves nearly double the maximum gradient strength of what the previous Connectome had. So it goes up to 500 millitesla per meter. And we were also able to achieve a very fast slew rate up to 600 Tesla per meter per second, which we're using to do faster diffusion and image encoding. Alongside the gradient system, we have, again, worked with Boris Kyle and his students to build several multi-channel array coils, sort of pushing the envelope of what we can attain within the tight space of this new gradient. So he and his team have developed a 64-channel ex vivo whole brain coil, which we are using for our validation studies, and a 72-channel in vivo head coil. So these coils are especially impressive because they fit within the 44 centimeter, basically, gradient diameter. So in order to get to such strong gradient strengths, we had to convert from a whole body gradient to a head only gradient. And then finally, to contend with the potential eddy current artifacts that we are going to encounter at such strong gradients, we had prepared in advance and basically built in field probes into the receive coil so that we could monitor the magnetic field in real time and then actually have the information ready to correct for those eddy currents as they are occurring. So the 3 Tesla Vita magnet is the platform upon which this scanner is built. This is a wide bore scanner and houses the hefty gradient coil, very much following the same kind of design characteristics as our original Connectome scanner, which also featured a wide bore scanner that was then outfitted with a gradient to fill the entire board. So the 3T Vita magnet was installed at the end of June. You can see here on the back, the gradient power amplifier cables, as well as all the water cooling tubes ready to receive that gradient. And this is the gradient coil in its completed form sitting in the factory in Erlangen. So this was done in partnership with Siemens. We've had a tremendous kind of collaborative effort over the last 5 years to bring this project to fruition. The Connectome 2 gradient coil is an asymmetric head gradient. It features a 3-layer design that's built on the same geometry as the Siemens 7 Tesla impulse head gradient coil. And that impulse head gradient was built through another Brain Initiative funded project led by David Feinberg with contributions from the MGH team. That gradient coil achieves a maximum gradient strength of 200 millitesla per meter and a slew rate of 900 Tesla per meter per second. So the gradient for the Connectome 2 features this stepped design and has shoulder cutouts, especially in the y-axis in the intermediate layer, to enable the shoulder to fit more subjects. The coil also utilizes novel direct cooling techniques, including these stainless steel tubes that are surrounded by the conductive copper filaments So it allows for cooling of the current as it's running through the wires and allows us to get to a higher gradient efficiency and operation at higher duty cycles. Here's the Connectome 2 versus the Connectome 1 specifications by the numbers. You can see that the bore size for Connectome 2 is considerably smaller than Connectome 1. A few people were asking me about this yesterday, so I dug up the numbers. The patient bore size is 40 centimeters. That fits within the inner diameter gradient coil of 44 centimeters. When we have a coil in there, for all practical purposes, the amount of space that we have is more on the order of 37, looking at the largest axis. The scanner achieves Gmax of 500, maximum slew of 600. If you take the vector sum, that goes up to 866 for Gmax and 1039 for slew rate. I'll also point out that the coil efficiency is actually better than comparable head gradient coils on the market. We had looked at the Magnus paper that Tom Fu had published about, from GE had published about that head gradient, and ours is substantially better than Magnus as well as the Connectome 1. Both are, the Connectome 1 and Magnus are more on the order of 0.3, ours is on the order of 0.4. And just to point out that the higher gradient efficiency of the Connectome 2 gradient coil allows us to use fewer gradient power amplifiers per axis. So in contrast to the 4 that were used for Connectome 1, we now have 2. And in the interim, Siemens has also invested in their amplifier technology and has developed more powerful amplifiers that produce 33% more power compared to the gradient power amplifiers of Connectome 1. So overall, it's a more compact, lighter, and potentially more energy efficient system, although I would say if you want to go for energy efficiency, this is probably not your instrument. So on top of all of the efforts that the Siemens team took to do torque and force balancing and also active shielding of the gradient coil, the gradient coil design was accompanied by very careful and systematic peripheral nerve stimulation modeling done by Matthias Davids and Larry Wald. And the idea, and I think the foresight that they had in sort of pushing forward on this gradient technology is that at some point, you may hit against the biological limits of what a human is able to sustain when you go to such high gradient strengths and high slew rates. So they use their PNS prediction framework, which relies on detailed computational modeling with embedded atlases of peripheral nerves from female and male body types, really kind of combining the electromagnetic and neurodynamic modeling to go through iteratively gradient coil designs and try to optimize the peripheral nerve sort of stimulation thresholds for this coil, sort of predicting in advance and then trying to essentially push out the PNS from the areas that would matter. So this is one quick example. It's a bit of a toy example, but I think Matthias produced this to really illustrate what they were trying to do. So here I'm showing on the left the gradient coil without the intermediate layer inserted. And you can see that there's a lot of peripheral nerve stimulation that would potentially be induced in the face. These are the thresholds, sort of the thresholds at which you would start to feel significant peripheral nerve stimulation. When we add in that intermediate layer, it essentially allows us to push many of these PNS hotspots more inferior so that they're kind of at the level of the shoulders or below, and to mitigate against the peripheral nerve stimulation that would be felt in the face. And so I'll go on to sort of the experimental measurements, but suffice it to say, these types of measurements have really made it possible for us to push the limits of the gradient and make use of more of the operational space afforded by this gradient coil. So after the construction of the gradient coil, Siemens did a volunteer study in which they measured the PNS thresholds in a group of healthy volunteers. And these plots are the summary of the population averaged peripheral nerve sort of stimulation thresholds for both the single axis modes and then combined axes. And for comparison, we're also showing here the threshold curves for the Connectome 1. So you can see here that the Connectome 2 gradient coil systematically achieves between up to 2 to 4 times greater PNS thresholds compared to the Connectome 1 whole body gradient coil. We're also kind of fortunate that we don't have to worry about cardiac stimulation now with the Connectome 2 coil because of its head-only design. So combined with the increase in gradient strength and slew rates, we have shown here that we can really get a dramatic improvement in the image encoding performance that's achievable while minimizing the degree of PNS that will be incurred when we scan multiple different subjects. So to go into a little bit more detail about these, the receive coils that we've had built through Boris and his students, we have now a 72-channel in vivo coil array, as well as a 64-channel ex vivo whole brain array. Both of these coils have their own dedicated local transmit coils. This was a design consideration that we incorporated it, so in order to try to optimize the shielding for the RF components as well as the gradient coil. And so we went with a split shield design in order to try to minimize the degree of eddy currents that would be incurred. So the 72-channel coil offers coverage of the brain, the cerebellum, and also the brainstem. The 64-channel coil incorporates kind of this bucket for whole brain specimens, which we plan to use for actually ongoing projects. It also has a temperature monitoring system. So this is where these air duct flows, these air ducts come in, and essentially we want to maintain a constant temperature within the whole brain during the very long acquisitions that we typically do for ex vivo whole brain imaging. So we can control the temperature and ensure the accuracy and reproducibility of the diffusion MRI measures that we're able to get, measurements that we're able to get during that time. So overall, down below, Boris and his student Mirsad came to Boston at the end of August. They did initial signal to noise ratio comparisons. As just one example, the 72-channel in vivo coil results in about 1.5-fold improvement in SNR over a standard 32-channel in vivo coil. And they're preparing these results for presentation at the ISMRM. So after all that, the technology behind the scanner, we were really excited to do our first in vivo human brain diffusion MRI scan. We did this on August 1st, the day after the system was turned over to us from Siemens. And as is usually the case, Bruce Rosen, our center director, volunteered to be the first person to go in. Bruce is very brave. He's also a little bit limited on time. So we cut straight to the chase and started ramping up the B value and Gmax pretty quickly. So at the B of 1000 scan, this is sort of similar to what Siemens is now marketing as a SEMA X. It goes up to a G of 150. SEMA X will go up to 200, slew rate close to 200. He was like, this is fine, keep going. So we went up to B of 4000, went up to a G of 300 millitesla per meter, slightly higher Gmax to what we slew rate to what we could achieve on the Connectome 1 scanner. And he was like, that was okay. I mean, I think I may have seen something in the periphery, but keep going. So then we went up to B of 10,000. And then after that scan, Bruce was like, I definitely felt something and I definitely saw something. There were these flashing lights in my periphery and I was feeling some tapping on my shoulders. When I told Matthias the results, he said, that's awesome. That's exactly what we want. Because in fact, I think the tapping that he felt was not so intense that it was causing pain. And it really kind of achieved our goals in terms of wanting to get close to the PNS limit, but not exceed it. In terms of the retinal stimulation, I think this is still a work in progress. And I know Larry and Matthias and others in the group are very interested in doing retinal nerve modeling. But I will say that not everyone has seen retinal sort of stimulation, which is what those flashing lights represent. It's a phenomenon known as phosphenes. And in fact, I think the majority of people have not seen them. So we're going to continue to monitor, and we will be doing volunteer studies to systematically evaluate these effects. The members of the group have been very excited and pushing forward to evaluate sort of systematically the capabilities of the scanner. First, characterization of signal to noise improvement for diffusion MRI. This was done systematically by Huang and Yixin. Huang is now faculty at Martinos. He came from NYU as a gift from Dimitri and Alison. He's been phenomenal. So he's really spearheaded the acquisition of the data on the system and guiding the postdoctoral fellows and sort of analyzing the results. So we can see here from the comparison of the images acquired on Connectome 2 compared to Connectome 1, systematically going up on B values, that we can achieve reductions in the echo time anywhere from about 10% at lower B values up to 50% with respect to Connectome 1. And in terms of the signal to noise ratio gains, we can see up to double the signal to noise ratio compared to Connectome 1. And it's, I guess, essentially infinite
based on this compared to the clinical scanner, because you can't achieve p-values of 40,000 on a clinical scanner. We've also started doing microstructural measurements as sort of a benchmark for how much better the diffusion resolution as well as signal to noise ratio looks on this new scanner. And I will say that the ultra strong gradients definitely improve the diffusion resolution for microstructural imaging. The application we focused on here is for axonal diameter mapping. So think back to the slide that I showed earlier with those 20 healthy subjects average. I think even with this, within the single subject, you can already see that the signal to noise ratio of the maps and overall the estimation of smaller diameter fibers is evident, as you can see from the color bars shown here. What's even more impressive, and I think this is really not shown to best advantage on just a single image on a screen, but rather best seen when you scroll through the images, is that when you look through the maps, you can actually start to appreciate variations in axonal diameter within individuals that kind of rival what you see when you average over multiple subjects. And so I would argue that that's actually probably going to be the benefit of this scanner for microstructure, is that we can now start to trust the measurements that we're seeing on the individual level and be able to appreciate individual variations in, say, axonal diameter or other microstructural properties that we were just at the cusp of seeing with our original Connectome scanner. And for me, that's the very exciting part. It's just one anecdotal example. In our original axonal diameter mapping studies in the healthy subjects, we did see this asymmetry in axonal diameter between the right and left hemispheres. And that was true, especially when we averaged over subjects, but you can sort of see it a little bit in individual people. Now that we've been able to replicate these results on the Connectome 2, I can more confidently say that this is actually probably a real phenomenon. At the time when we observed it, we thought, well, maybe this is an artifact. We even had people go into the scanner, both supine as well as prone, to make sure that it wasn't like a coil issue, but I mean, we didn't think it was a coil issue. So anyway, this is, it's very exciting. And I think it will usher in this sort of the advent of precision neuroscience and our ability to look at individual variations using these more powerful systems. We've been able to push the sensitivity of diffusion MRI. These were results that were just acquired last week. In addition to pushing the diffusion resolution, we can also use the improved signal to noise ratio for diffusion to push the spatial resolution of diffusion MRI. And this is a very important goal and effort that we want to sort of achieve for both in vivo as well as ex vivo imaging. So Bilgic in our group has also implemented a version of the G-slider technique that we had originally worked on together at Cowan Setzenpop on the original Connectome scanner. And we have a working version of the G-slider sequence on the Connectome 2 scanner. We're really kind of looking to optimize this further, but you can already see the improvements that you can gain over the Prisma scanner, even at low B values. And then as a quick preview of what's to come, we have implemented the field camera monitoring on the scanner. Those field probes are now integrated into the coils, and they have been successful in terms of allowing us to really measure the field in real time. And so these are results from a recent paper that Gabrielle Ramos-Jordan and our group published, showing that with dynamic field monitoring, incorporating the sort of perturbations in the magnetic field coming, say, from eddy currents in the image readout, you can dramatically clean up the artifacts that you see from such, especially in segmented acquisitions like this 3D multi-shot diffusion weighted EPI sequence, to get estimates of, say, orientational structure that are less biased compared to what you would get if you didn't incorporate these measurements. And so taking these types of technologies together, we're incorporating them into a project, a very large scale project that Anastasia Yandicki, my friend and colleague at the Martino Center, has spearheaded. This just started. It's a Brain Initiative UM1 comprehensive center for doing large scale imaging of neural circuits. Essentially, the goal is to map all of the connections in the human and non-human primate brain at the axonal projection level. And to do this, as Camille was alluding to, we really have to bridge the scale. So we have to use multiple different kind of modalities and technologies and to try to piece those together. So from the monkey side, we're going to be doing tracer injections led by Suzanne Haber at Rochester, and then imaging those brains with our small bore scanner, which achieves a maximum gradient strength up to 616 millitesla per meter. Those will serve as a guide for sort of parallel experiments done in ex vivo human brains done on the Connectome scanner using that ex vivo coil. And then following that, we're pursuing several technologies for imaging at the mesoscopic scale, including polarization sensitive OCT, as well as hierarchical phase contrast CT, and then going down even further to the micron level with paralleling this with micro CT and light sheet microscopy, working with Elizabeth Hillman at Columbia. So we look forward to kind of sharing the results from these this ambitious project in the next few years. And so the last kind of topic, well, what's beyond this? What's Connectome 3? And so this is a quick project that I'll just highlight. This just started, and I'll point you to Jason Stockman, who's our fearless leader on this, but we have essentially undertaken an array for reception, encoding, shimming, and stimulation. This is essentially a crazy idea that we had a few years ago. Essentially, to get to much stronger gradients, we realized we might have to take the gradient coil out of the bore of the magnet. And so we're now going to be using the magnetic fields generated by transcranial magnetic stimulation probes to generate much stronger amplitude gradients near the cortex for cortical diffusion, as well as for image encoding. And so talk to Jason if you're interested, because we have job postings available. And yeah, with that, I want to acknowledge everybody, especially at the MGH, who've participated and contributed to this immense effort, and also to the broader Connectome 2 team, especially our colleagues at Siemens, for all the work they've done on this. Thank you.